Now, we've set these up on purpose so that we have 2 speakers each time, one from East Campus typically and one from West Campus. Today is a great example of if we just gave you one, if we just gave you Chris Gobler, you'd be very lucky. But you have Chris Gobler, you have Sharon Nackman, and we have also an opportunity to think about some really interesting ways in which their talks will be very different, but also have some common themes. And so one of the things we'll want to do is think about where those are and what that means at a university that I believe doesn't just say we are interdisciplinary or that we care about that, but actually shows that in the decisions we make, in the ways that we work together, and in the ways that we are setting out the path for the university. So as we start to begin now, I'll just want to say quickly, Chris Gobler is a SUNY Distinguished Professor in SOMAS, the School of Marine and Atmospheric Sciences, and he's an Endowed Chair of Coastal Ecology and Conservation. He's also the feature model in the strategic plan, so if you want to take a look at that, he is dead center on the cover. So if you forget what he looks like, you can always pull out your strategic plan, because I'm sure it's not sitting on a shelf, but it is right at your desk, helping you think about how we are moving forward as a university. Chris received his bachelor's degree in biology from the University of Delaware. He later received his master's in marine environmental science and his Ph.D. in coastal oceanography from Stony Brook University. So this is a case of our own now becoming one of our true greats. He started as an adjunct professor here at Stony Brook in 2002, an associate professor, and now, as I mentioned, is one of our SUNY Distinguished Professors. I will refrain from saying more because we have one of our great deans, Paul Shepson, who also is a SUNY Distinguished Professor and faculty within the Atmospheric Science Division of SOMAS. I just want to say a few words about Paul. He's an atmospheric chemist. He's now in his 2nd term as dean. What I really love is he's been at every single one of these. And Paul is someone who really deeply cares about students, faculty, and staff within SOMAS, but the university more broadly. And when we have that deep passion for our discipline and our area, but we also are seeing the bigger picture of what we can do together as a university, it's truly great. And, you know, I think he's got like this little bit of humanist in him. He is someone who truly deeply cares about scholarship, academia, and what our responsibility is to our students, to each other, and to our society. So with that, I will bring up Paul, and he will get us started.
So thank you for that introduction, Carl. I really appreciate it. And we'll exchange compliments. So Carl is very good at an intent in complimenting the person that he's introducing. And he does a fantastic job with it, and it makes you feel humble at the same time. So thanks for the opportunity, Carl, for me to also do my own presentation. Carl loves to hear me speak, so I appreciate it very much. So I am delighted to introduce...
We'll cue the music if I have to.
Yeah. I know what that music sounds like. So I'm delighted to introduce Chris Gobler to you, and I'm going to start at the beginning. And I guess I mean just Chris's beginning. So Chris was born in Port Jefferson, New York, so you might have heard of Port Jefferson. He went to high school at Ward Melville, grew up in South Setauket. Eventually, after graduating, he went to far away, one of his main trips far away from Long Island. He went to University of Delaware, to get a degree in biology, a BA in biology, and then he came back to Stony Brook and did a master's degree in marine environmental science and a Ph.D. in coastal oceanography, which Carl already said. It was then at the time the Marine Sciences Research Center, and I'll come back to that. But this is when Chris first learned to go deep in the coastal marine sciences, as we like to say. He then took a position as assistant professor at Southampton College, where he got tenure. And at that time, the Southampton Marine Science Program around that time became part of SOMAS, where Chris is now director of academic programs at the Southampton campus. As Carl said, he has an endowed chair in coastal ecology and conservation. He's also director of the New York State Center for Clean Water Technology, and I'll come back to that. So Chris has a monstrous, very active, very vital research group with a pile of grad students, something like 15 at a time, several postdocs, and lots of undergraduate students. So when you go visit the Southampton Marine Science Center, you just see a hustle and bustle of activity. It's a sort of an intellectual center, very exciting place to be. In his research, Chris focuses on coastal plankton ecology and harmful algae blooms and their impacts. He connects molecular level understanding of those events and the linking of genomics to environmental conditions. His study of harmful algae blooms has investigated the role of both macro and micronutrients in the genesis of the HABs and their toxicity, and also the impact of climate change. Chris is a very prolific researcher. He has on the order of 250 peer-reviewed publications and has been cited 27,000 or so times, speaking to the hard-hitting character and impact of his work. Quoting a colleague in the field from a couple years ago, he said, "This is how he made his career, a careful and deeply understood assessment of the sequence of choosing great projects, performing the science, and preparing communications." But I want to say that Chris is about much more than just doing good science. He works really hard to connect the development of fundamental understanding of the problem at hand to solving real-world problems. And in so doing, Chris personifies the SOMAS motto, making scientific research count. He, in fact, is so committed to this that he created the New York State Center for Clean Water Technology, which, among other things, helps Long Island and Long Islanders improve their human waste-related nitrogen runoff from our relatively poor septic systems, and in so doing, helping to mitigate future algae blooms and protect human health. So Chris is the real deal in doing hard-hitting fundamental science and then connecting that to real societal problems. We have big ones here on Long Island in terms of managing our waste. So I could go on, but then you'd be listening to me and not Professor Gobler. So let's welcome Chris to the stage.
Okay, well, thank you so much for the introduction, Paul and Carl, and thank you for coming. It's an honor. And I have this really long title here. I don't know where that title came from. I went to my Google Calendar and looked up what I had in there, and that's what I had. And I thought it was actually terrible. And so I've actually changed it. Maybe there's some good science words in there. And when I was trying to think of how to change it, I thought about how I got here. And so anybody who's a fan of J.R. Tolkien, maybe you've heard of Bilbo Baggins wrote the book There and Back Again, A Hobbit's Tale. And so I've altered that a little bit to There and Back Again, A Seawolf's Tale. And Paul gave the introduction essentially showing that I am a local. And so this is me here. You can see West Meadow Beach. And it turns out my parents, my mom and dad, are actually here. They didn't have to go very far to get here, as you can imagine, coming again from South Setauket, where we grew up. And so I'm going to use this sort of local narrative to start things out here, and play it over the narrative of what's happened locally as well. And so, we started Suffolk County, the start of the century, as a fairly actually rural location. My parents tell me about what the roads were like when they first came out. I think there was just one light on Nesconset Highway, as I understand. And it was in the 1960s that Stony Brook came to this particular campus. It was the next decade that I was born and raised locally. It was the decade after that I went to graduate school here. And for the most part, I've been faculty here since then. And just for perspective, you actually needed to see where I actually was. You know where the university is, I believe. Let's see. Well, you can see where it is. Maybe I'll figure out how this pointer works. So there it is approximately. That's exactly how far my parents live away from where the university is. And so growing up, I actually remember a scene like this where the hospital was actually being built. And I confirmed with my father the other day that this was actually true. We had a tree, very, very tall tree in the backyard. I could climb up, and from that tree, if I looked far enough, I could actually see the hospital and remember the hospital going from 1 to 2 buildings all the way back in the 1970s. And it was also during that time concurrently that things were changing on Long Island in other ways. And so specifically in the 1970s, as you can see in the 2 images in the upper left there, Long Island had the largest hard clam fishery in the United States. 2 out of every 3 hard clams eaten in the United States came out of Great South Bay. You can see some of the amazing pictures and all the boats lined up in Great South Bay. There's the Robert Moses Bridge and the incredible yields that came from there. And it was a way of life on the South Shore. We had family members we knew who were clammers and other people who lived in the water and enjoyed the water. But things changed drastically thereafter. This fishery entirely collapsed. And I remember learning about this and coming to the question of why is this? And the answer was brown tide. And this is what a brown tide would look like. And then the question I had thereafter was, well, why do we have brown tides? And nobody had an answer. But it was something that kept sticking with me over time. Why are these brown tides happening? As I was finishing up my undergraduate degree, I wanted to answer that question. And well, it turned out there was only one institution that was actually looking into it, and that was Stony Brook. And that's actually what brought me here for graduate school. But to back up very briefly, what are brown tides? Well, they're phytoplankton, microalgae. For those of you who don't know, of course, they're the base of all marine food webs that supply all of our fisheries and actually supply the planet with half of the oxygen that we breathe. But as it turns out, there are thousands and thousands of species. There's several hundred that can form these things we call harmful algal blooms, like these brown tides that can have a negative effect on human health or on ecosystems. And they're a widespread concern. This is just one type of harmful algae, the type that causes what's known as paralytic shellfish poisoning. That's a neurotoxin that can lead to the intoxication of humans and death at very high levels. And you can see it's a global phenomenon. And this is just one of, again, dozens, if not hundreds of these species. With regards to brown tide, you've seen what the water looks like. The cells are very tiny. You end up with over 1 billion of these cells in just a liter of water. It can obscure the water, and it is toxic to some forms of marine life, particularly shellfish. We knew these species were around in the Northeast, or this particular species that caused these blooms, the brown tides, in the Northeast in the 1980s, as I mentioned. Through the 1990s, they were discovered to have spread further south in the east coast of the U.S. A sister species was found in the Gulf of Mexico, and this was even found all the way across in South Africa. Since then, with collaborators in China, we discovered this same exact species was there. And my lab also discovered the spread of the sister species south into the Caribbean and Florida thereafter. So these are really globally spread phenomenon. And coming back to the US, we find the organism all up and down the east coast of the US. And blooms that have had negative impact on ecosystems have occurred between Rhode Island and as far south as Virginia. And beyond the hard clam, locally here in New York, this harmful algal bloom was actually also responsible for the complete collapse of the bay scallop fishery, second biggest fishery in the, of that species on the East Coast, and the loss of eelgrass habitats, which were a critical habitat for many, many fisheries. And again, one of the big burning questions of my research, actually both starting in graduate school, but then even as I began as a faculty here, was how does this happen? You know, typically there was a seminal paper published in the 1960s called The Paradox of the Plankton, where theoretically there should be hundreds, if not thousands, of species coexisting at once. But during a brown tide, the whole community changes, and it's just brown tide that's having that negative impact on the ecosystem. So as I began my faculty career, it was at the turn of the century, and technically, as I teach in my classes, this century has been that of the genomics era. Kind of like the slide that is from collaborators in China, and specifically the Beijing Genome Institute. I like the title. It's we are now in genomics era. But I like more the time frame because essentially what it shows is that in the beginning of this era here, you can see the first human genome was only published in 2001. And you know, the first, I think, algae was just a few years thereafter. So to really crack the code on what was causing brown tides, one of the big steps that we took collaboratively with the Department of Energy and many other scientists was to sequence the full genome and understand, you know, what makes this organism tick, you know, what's in its genome. And again, that was a pretty novel question at the time. This was the first harmful algal bloom to have its entire genome sequenced. And I won't go through all the details, but just to go through some of the highlights. At the time, we had a lot of other genomes to compare the brown tide to. So some of the insights were specifically that even though this is a very, very tiny cell, it actually had a larger genome and more genes than all the phytoplankton species that we knew it was competing against. So sort of a deeper toolbox to draw upon. And within that toolbox, it also had many, many genes that allowed it to adapt to low light conditions and to using organic matter. And also we had some sense that it also was able to defend itself against predators by making specific bioactive compounds. And I won't go through the details here, but just to point out, this is from that paper specifically looking at Oreococcus, and to the right of it, all of the competing species. So there's the brown tide and the competing species. And so on the top, more light harvesting complex genes. The middle figure there showing had more genes breaking down organic nitrogen. That becomes important momentarily. And also more enzymes that had metals in them, specifically a record for the most what are known as selenoproteins. Nick, I thought you might like that particular line there. But a genome just tells you the potential of the organism. It really doesn't tell you what's going on. But you know, we had an incredible opportunity, though, because of these blooms happening locally, to essentially go into these brown tides occurring and understand over time what parts of the genome are active during which phases of the bloom, to understand really physiologically what's happening to these cells, and essentially why are we getting these blooms. And I'm showing you what a harmful algal bloom looks like with regards to the data set, and specifically this is one you can see initiating in June, peaking in July, and then continuing on and collapsing ultimately late in the summer. But this is like a 6-week period, and this actually was a great data set because it allowed us to capture what we call the initiation of the bloom, the peak of the bloom, and then the demise. And specifically what we wanted to do is to understand, as it went through this trajectory, what genes were being expressed that might be an indication of what's driving the bloom. So firstly, just to look at what else was in the water and sort of confirm what I was told you before, there's that same progression of the bloom on the top. On the bottom are mapping different transcripts during the bloom, so there's the RNA that's being expressed to different organisms. and essentially saying that if you look at the bottom graph there, the majority of the RNA in the water was coming from Oreococcus, or it really was the dominant species. In some cases, 90% of the RNA out during the bloom. There's a lot to this figure, and I won't spend too much time on it, but essentially this is using what we'd call global transcriptomics, looking at all the different gene sets that are being expressed over the course of that bloom. And again, what we want to do is break that bloom down into 3 parts: the initiation, the peak, and the demise. And specifically, in addition, we could figure out how fast it was growing at these different points in time. So in the very beginning, it was undergoing explosive growth. In the middle, it was sort of in a maintenance phase, and then completely collapsing. And so if we look at the different genes that are being expressed at the different times, we can see in that initiation phase a distinct signal. So specifically the genes associated with making lots of DNA and making lots of amino acids to make sure that this population was ready to grow very, very quickly. And then when the bloom became very, very dense, the water is very, very turbid, we see that entire gene set change. And specifically in that maintenance phase, there's carbon fixation going on, not any surprise there, but using a lot of organic carbon by the cells in order to maintain themselves, and breaking down fatty acids to maintain themselves. And then finally, at the collapse, we see everything change entirely. So a complete downregulation, sort of indicating a death spiral for the bloom. And then we targeted in on specific genes related to specific nutrients, specifically nitrogen and phosphorus. And what we saw is that maybe in the beginning of the bloom, we had a hint that the bloom might have not enough phosphorus, but definitely during that peak of that bloom period, a lot of genes related to nitrogen. So the first hint that nitrogen might be important here. To test that further, we conducted an experiment where we collected water and simply amended that water either with nitrogen or phosphorus or both compounds to see how that would alter the dynamics and growth of the bloom. Just looking at the growth response, we saw a strong response from nitrogen, a little bit more of a muted response from phosphorus, and then the biggest response from both of these genes. And I won't go through the details here, but this is a non-dimensional scaling plot just showing how different the transcriptomics or the transcriptomes of Oreococcus was, comparing that control condition with nitrogen, and you can see the other combinations somewhere in between. And again, looking globally at the full transcriptome elicited in the different treatments, the nitrogen and the phosphorus or both, and comparing it to the control, we looked at genes that were either being upregulated, that's sort of in the bluish purple color, or downregulated in the orange. And so what we could see during this experiment is that when there was more nitrogen, all these important pathways for the growth of this organism were being very strongly upregulated, sort of giving the signal that nitrogen might be the key player in driving this bloom. But unfortunately for what's happening here in Long Island, that occurrence of brown tide in the 80s was just the tip of the iceberg when it came to these harmful algal blooms. Since that time, my lab has discovered many, many other types of harmful algal blooms that have emerged all across Long Island, on the North Shore, on the South Shore, of the East End, some of which are damaging, like Oreococcus, others of which are actually toxic to humans, and that PSP that I showed us globally, well, we now know that's here on Long Island as well. And it's widespread. This is a map just from last year showing the widespread occurrence, again, of these events all over Long Island, the South Shore, the North Shore, et cetera, all different species. And I won't go into the details, but again, nitrogen has been the smoking gun in many of these events. We've been able to show that both these harmful algal blooms can become more intense, but also importantly, many of the toxins they make, the biotoxins, neurotoxins, gastrointestinal toxins, are nitrogen-rich compounds. So with more nitrogen in the water, they can synthesize more of these compounds and actually not just become more intense, but also more toxic. And then just stepping away from harmful algal blooms, we've looked at other things that can be changed by overloading of nitrogen. And that's an example of this here, a study looking specifically at how excessive loading at nitrogen affects not just dissolved oxygen, which had been known, frankly, for decades, but also the pH of the water and something known as acidification. And really the discovery with this paper that what we've known about for decades, hypoxia, is not an issue in and of itself, but it's also coupled almost always with acidification. And I'll just point out, this is a cruise from Long Island Sound. That area in red is what we'd call the dead zone in Long Island Sound of low oxygen. It's accompanied by pH that's very, very low. Sometimes oceanographers worry about ocean acidification because they say, well, the ocean might get down to as low as a pH of 7.7. If you look at the scale here, this is a pH of 7. And in fact, in some cases, a pH that's almost is below 7, so actually acidic. So these are very low and levels of pH and very high levels of CO2. So this is from a cruise through Jamaica Bay, essentially showing that when you get to the low oxygen areas, you can have levels of CO2 of 4,000 parts per million, an order of magnitude more than what we have in our atmosphere now, and well beyond what we expect in the future. And that's happening now, and the root cause is that overloading of nitrogen. And that's why we've been able to talk about the idea that current policies to protect fisheries that are only considering oxygen are actually not adequate. You know, there's actually the only defensible water quality metric in the Clean Water Act that has true metrics that can be defended is dissolved oxygen. And the ones for pH are totally inadequate. And if you look at the way they're stated, they really need to be reconsidered. And that's sort of what we outline in this paper after discovering the co-occurrence of that hypoxia and acidification. And of course, that acidification has incredibly important biological implications. This study was looking at how those increasing levels of CO2 would affect the growth rates and survival rates, specifically of the larval stages of shellfish, those hard clams and bay scallops that are so important locally. And essentially what we're able to show is a dose-dependent relationship. More CO2 leads to lower survival. And the concentrations here are really important. So, you know, we started when this paper was done, actually, almost 15 years ago. That level of CO2, 390, well, that's what the atmosphere was. We're now at about 430. So things move fast when it comes to CO2. The 250 was what we had during the pre-industrial era. And so this paper actually showed that very simply the change since pre-industrial times to today is enough to elicit a depressed survival rate in these bivalves, and that going forward, it's only going to get worse. And in that same paper, we just showed beyond the survival, what the images look like, what the bivalves look like at the end of the experiments, and essentially you can see the very, very clear reduction in the size of these organisms. They're also thinner. And this image actually was used in the third National Climate Assessment and actually used by the Obama White House when they published a paper called The Challenge of Ocean Acidification. And of course, another attribute of beyond acidification of climate change is warming. And we know that last year was the warmest year on record on this planet. And we also know that right now, in 2024, our world oceans have never been warmer than they are at the present moment. And in fact, this is data the University of Maine administers. You can see 2023 was off the charts. 2024 is off the charts even further, because all those squiggly lines you see below that are the prior years of data. So we're kind of in uncharted territories, and a lot of people, scientists, don't actually even understand why. But this has important implications for harmful algal blooms as well, because as waters warm, organisms want to stay in their thermal optimum. And so what they tend to do, therefore, is migrate towards the poles. So in this paper, looking at another harmful algal bloom species, we're able to show that the warming that occurred from 1982 onward, I think this paper was published in 2017, essentially has led to opening a new niche for this toxic organism. This is the one that makes that PSP, paralytic shellfish poisoning, and that it's the length of its bloom season, how long it can be growing and forming these harmful algal blooms, greatly expanded in several regions across the North Atlantic, all across Northern Europe and in the Northeast US and also in Canada. So we've got lots of things occurring concurrently. We've got climate change going on, but very importantly, not forgetting, the nutrients and the nitrogen are really a prime driver here that's then acting additively and even synergistically with climate change. So if this is the cascade that's getting us to our problems, the question is, how do we halt this cascade? And in many ways, it's the nitrogen loading. That's what's stimulating the harmful algal bloom. It's what's leading to the hypoxia, leading to the acidification. The nitrogen loading, I showed earlier what's happening with the population of Suffolk County. And as that growth has occurred, the nitrogen that's in the groundwater below our feet has been progressively increasing. It's increased 60% since 1980. And this groundwater, one, we're drinking, and I don't have time today to talk about the public health implications of high nitrate in drinking water, although I will mention that EPA is currently reassessing their standards about that. But I will mention that what we don't extract to drink in the groundwater discharges to surface waters. And of course, here on Long Island, we don't have major rivers. The main source of fresh water that dilutes our marine waters is groundwater. So this high nitrate groundwater is entering our surface waters, and that's increased a lot over time. And the reason it's increased is because of what we call on-site, residential on-site wastewater systems. So it's maybe it's a dirty secret, that 70% of Suffolk County is not hooked up to a sewage treatment plant. Thankfully, this campus is, but 70% of the homes aren't, and they're just draining into that aquifer that we're drinking from, but also from considering coastal zones. That means that's the source of that nitrogen. That's sort of settled science. There have been many, many studies that have shown essentially that the source of the nitrogen that's in our groundwater is primarily from this on-site wastewater draining from individual homes. And that wastewater, therefore, obviously you can imagine, is very, very potent. If you add up Suffolk County and Nassau County, there's 400,000 of these systems to deal with. And so that's been a focus of something called the New York State Center for Clean Water Technology. I didn't found the center entirely, actually, just to correct something that Paul gave in the introduction. It was founded by a former governor and other politicians, all of whom are now out of office. But the center remains, and the motto of the center, or the mission of the center, is to harness science to engineer clean water for the protection of public health and the environment in New York and beyond. And one of our big focuses has been, can we have and can we build a better on-site septic system? And we think we have something called the nitrogen-removing biofilter. It's incredibly simple. It's based on the premise of having a sand layer that's underlined with a layer that has sand and wood chips combined, and create something called coupled nitrification and denitrification. So very, very simple, and above that is simply a drain field, which is the way that most on-site wastewater is dealt with across the country, but not here in Suffolk County. And in exciting news, the system that we have developed, that we have two different types, are actually outperforming every other system that's being tested in Suffolk County. You know, backstory, Suffolk County passed something called Article 19 about 7 years ago that had a very rigorous program for upgrading systems, testing systems, and the two systems that we've created have nitrogen effluent better than any other system, including several that have been found all across the world. So we're very, very happy about that, and one of the systems has been provisionally approved by Suffolk County. We expect the other one to be provisionally approved in just a couple of months. And of course, we're drinking our groundwater, and our wastewater is going into our groundwater. There's more to remove than just nitrogen. And so a happy outcome of these nitrogen-removing biofilters, if I just go back, is that sand layer, as it turns out, is a magical layer biologically, and that there's all sorts of bacteria that love to live there that break down things beyond nitrogen. And so this is just a list here of more than 24 drugs, pharmaceuticals, personal care products, industrial solvents that we don't want in our drinking water, but are removed at fairly high rates within the nitrogen-removing biofilters. And even if these rates in some cases don't look high, in every case it's higher than you'd get out of a sewage treatment plant. And so again, as I mentioned, these systems have been provisionally approved in Nassau and Suffolk County, and there are grants available to install them, so we have great hope that this will be a great solution going forward. And then the very final thing I'm going to mention is things that we're doing and have done in the water to also protect these coastal ecosystems. We need to upgrade these septic systems, but if you don't know, groundwater takes a long time to travel from one point A here out to being discharged. In some cases, decades. So there needs to be, in parallel, other approaches for protecting surface waters. And one thing that we've done on that front have been using filter feeding bivalves. The awesome power of filter feeding bivalves. One oyster can filter 250 liters of water in a day. And this is just an experiment showing a 300-gallon mesocosm. And if you can see on the left, at the bottom of that is a container with, I think, just about a dozen clams. And on the right is a container that didn't have those clams. And that's a brown tide right there. So these clams can filter the water and improve them, the water quality conditions, and prevent harmful algal blooms. So without having those, you're in the situation we're in today. With them, the idea is maybe we can turn this around. And so we did this in something I'm going to show momentarily called the harmful algal bloom, the Shinnecock Bay Restoration Program, where we didn't just want to plant clams to filter the water. We wanted to use their awesome filtration power with their awesome reproductive power. As it turns out, a male and female clam can have 1 billion offspring in a year. And so we planted 5 million of these clams with the hopes that they create dozens and dozens, millions more. So you can see the 5 million we planted. This is all in western Shinnecock Bay. And we had great success with this. I'm going to zip through it. You can read the paper for more details. But over time, we saw the population grow by more than 20-fold. And if you look at the clams that were created in Shinnecock Bay and you look at the colors there, these are all little tiny clams. So we know they were created by those spawner sanctuaries because they're new recruits. And it totally changed the trajectory of the fishery. We didn't anticipate this. We just really wanted to increase the total population. But what we saw instead is that the landings of hard clams that Bay were able to pull out increased. We started the program exactly in 2012. And so you can see what happened thereafter. There are now more clams being pulled out of Shinnecock Bay than Great South Bay, despite it being 10-fold smaller. And all these clams that were created are filtering the water at a faster pace, and it actually led to the end of brown tides in Shinnecock Bay. And with that clearer water, an expansion of the seagrass beds in Shinnecock Bay as well, over 400,000 square meters. So again, the idea being these clams, by filtering the water, are creating habitat for seagrass and also getting rid of those harmful algal blooms. And so, the motivation for my work was to get into figuring out brown tides and the impacts they had on clams. And, maybe I finally landed in a place where we're starting to reverse those trends. Anyway, I think I'm out of time. I thank you very much for paying attention.
We have some time for questions.
Thank you. 2 quick questions. Can you eat the clams that have serviced in this to remove the brown algae?
The answer is ask the DEC. So, I talked about this with the people before. Sometimes I give presentations and people never want to eat seafood again. But the fact of the matter is there's incredibly great regulations on shellfish that are made by the federal government, something called the Interstate Shellfish Sanitation Commission that's promulgated down to the states. Bottom line is any bivalve that's eaten in New York State or anywhere, there's essentially a whole, not quite a blockchain, it should be a blockchain, but a blockchain of information stating where it came from, when it was removed from that particular location, and who harvested it. And there's every single bottom of the bays of New York, from New York Harbor all the way out to Montauk, are specifically designated as open or closed. And so with regards to are they clean to eat after they filter the water, if they're filtering toxic algae, the answer would be no, but the DEC would have then closed those shellfish beds, although over time they can do something called depuration, that is getting rid of the toxins. But if they can actually, that's how they eat normally. So in other cases, they're filtering the water, as long as they're not toxic algae, that's what they do. And actually, that's what actually makes them nutritious. The algae actually have some of the things like omega-3 fatty acids, for example, that are made by the algae and then concentrated in the bivalves.
Also, what role do the fertilizers that we put on our lawns, all that nitrogen, is that a significant contributor to the nitrogen?
It is, but it's number 2. It comes in second. In fact, number 2 is number 1, I guess. But it is the second biggest source of nitrogen to surface waters.
Is it enough that we shouldn't fertilize our lawns?
this certainly reducing, in some areas, we need reductions of nitrogen loading that includes, upgrading the septic systems and reducing the fertilizer. And, in some ways, reducing the fertilizer use might be easier than reducing the septic use. So, I think attacking both makes sense.
Hello. I'm the Stony Brook student. I actually wanted to ask, this is a little more of a fun question. I know that Roth Pond has a lot of algae blooms. Is it a similar one to what you're talking about? I know marine and kind of like the pond is kind of a different ecosystem, but they do both experience algae blooms, and that's why a lot of students are told not to swim in it. But I also hear that they also clean it sometimes. Is it a similar case?
Yeah, so there are algae that grow in Roth Pond. In fact, as it turns out, my lab's monitoring Roth Pond on a weekly basis, getting ready for the regatta this month. And yeah, so in some cases they're different. They're similar in that some of the ones that show up, particularly in summer, can be harmful to the ecosystem. And yeah, but we're on the case. We're monitoring that, and so far things look good for the regatta for 2024.
Chris, nice talk. So you mentioned that there are many hundreds of species of algae out there, and most do not produce harmful toxins. So I don't know what percent do, probably less than 10% maybe.
Yep.
Right? And and they're expensive to make for these species.
Yeah.
And the question is, what advantage is conferred upon those species that actually go to the trouble of spending all that energy building making those toxins? What do they get out of it? And if we understand more the advantages that are gained, we might be able to figure out ways of countering the production of toxins by these species that they're not always toxic. They just occasionally produce toxins.
Yeah.
Could you comment or speculate on
Sure.
why
Yeah, I used to call that the $64 million question, but with inflation. No, I'm just kidding. It turns out, actually, in the last decade, there's been some pretty good breakthroughs showing that at least some of those compounds, saxitoxin, domoic acid, seem to be upregulated by predators and therefore serve as a predator defense, which sort of makes sense. And you're saying totally, if you're going to invest in doing that, there better be some sort of payoff.
Right.
So that seems to be the case in several instances. There's exceptions, and people are still trying to figure out those exceptions.
Yeah.
Still time. So I did forget to say that Mr. Long Island here is the scientist of choice for the media about these issues. You know what a great communicator Chris is now. But we have a challenge, and to me it's about all of the conversation now about geoengineering as an approach to dealing with climate change. The most common method being injecting sulfur in the stratosphere to decrease incoming radiation. But that approach ignores the role of rising CO2 and ocean acidification. And in fact, that approach will sort of be a distraction and probably cause CO2 to go up more. So this particular message about ocean acidification is not getting through. So I'm wondering what you think about that and how we deal with that.
Yeah, it's a very good question, and actually, there's been in my mind a fascinating arc on this front. In that, I'm going to get the years wrong, but somewhere in the turn of the century, there was a meeting that started that was called "Oceans in a High CO2 World." And in the very beginning, the focus was on how do we sink more carbon into the ocean. And then when people discovered that actually doing that acidifies the ocean, then the meaning totally changed, and the focus was entirely on ocean acidification. But now it seems the pendulum's swinging back again. And so, I mean, you're very right. There's all sorts of plans. How do we sink carbon into the ocean? But, you know, the fact of the matter is, the more carbon that goes in, the more we're acidifying that ocean as well. So, yeah, it's tricky, and I actually don't, I understand why the people went away from the sinking of carbon to mitigate the CO2 problem. I guess there's a financial reason for it swinging back, maybe. A lot of investment, but anyway, it's something to think about, so.
Let's give another round of applause.